Hello everyone, I am Donia Hussien. I'm a master's student at Ain Shams University in the Faculty of Science Biochemistry department. Today I will present my project that's entitled with two novel Streptomyces strains for paracetamol-traces bioremediation with chemo-informatics safety examination of the potential biologically active biodegradation products. This project is under supervision of Professor Dr. Magdi Mahmoud, Professor of Biochemistry, Faculty of Science, Ain Shams University, Professor Dr. Nadia Hamdi Al-Hafni, Head of Biochemistry Department, Professor of Biochemistry, Faculty of Pharmacy, Ain Shams University, Associate Professor Ahmed Shukri Abderazik, Associate Professor at Microbial Chemistry Department, National Research Center. During the period of COVID-19 pandemic, the environment was affected with pollution and resource depletion, like pollution with pharmaceutical compounds like paracetamol, disinfectant misuse, and microplastics pollution. Number two, increased the waste, especially the medical waste, and also electronic wastes. Number three, the biodiversity and ecosystem disruption, which mean reduce the conservation efforts, wildlife disturbance. Paracetamol is one of the emerging pollutants that can affect the environment and the ecosystem. That's because it's one of the over-the-counter drugs that's used in a huge amount during the period of COVID-19 pandemic, leading to bioaccumulation of paracetamol in the environment, especially in the wastewater, groundwater, fresh water, and also it reached our food. Papers proved that unmetabolized acetaminophen or paracetamol can inhibit ribonucleotide reductase enzyme, leading to chromosomal aberration, and also long-term use or long-term exposure to paracetamol with even low concentrations can lead to hepatotoxicity. The annual global consumption of paracetamol ranged from 2,300 tons to 5,200 tons per year, and the highest consumption is in the developed countries compared to developing countries, and it's expected that the consumption of paracetamol will increase during the coming period. That's because the increase in the population, the healthcare problems, and also the aging population. And this graph explains the huge consumption of paracetamol during 2020. That's because pandemic COVID-19. Because of the huge consumption of paracetamol released to the environment, especially from wastewater treatment plants, hospitals, pharmaceutical manufacturing plants, and also human disposal of medications. Around 50 to 90% of consumed paracetamol ends up in the wastewater. And also studies have proved that paracetamol was present in the water bodies worldwide, in rivers, lakes, even drinking waters. This project aims to find an eco-friendly, low-cost, reliable, easy and sustainable technique to get rid of paracetamol bioaccumulation in the environment. This technique is called bioremediation, in which we use microorganisms to degrade paracetamol and give us more safe degradation products. So, during this work we have isolated bacterial strains from different locations in Egypt, and then we identified the degradation product by using chemical methods like GC-MS and LC-MS, and after that we have tested the safety of this product compared to paracetamol itself. to make sure that the degradation products are more safe than paracetamol itself. This graphical abstract represents all the steps of the project, starting with biodiverse samples were collected from different locations in Egypt and then processed and cultivated on enriched media. And after that, the isolated bacteria was cultivated on mineral salt media. This media was used, this media contain only minerals, and also we have added paracetamol as a sole carbon and nitrogen source for the bacteria with concentration of 500 milligram per liter. And after that, growth of bacterial colonies. This colonies was incubated and then at 30 degrees Celsius for 7 days in static and shaking flasks. Then the cultures were filtrated using filters, and the filtrate was extracted by using equal volumes of ethyl acetate repeatedly. Then the organic phase was collected and evaporated, giving us crude extract. This crude extract was used for the identification of the metabolic products by using JC-mass, LC-mass, and HPLC to check the metabolic product of the bioremediation process of paracetamol. And then the isolated bacterial strains were identified using phenotypic identification by transmission electron microscope and also genotypic identification by 16S rRNA sequencing. The crude extract also used for toxicity experiments like acute oral toxicity and also in vitro toxicity on cell cultures, HBG2 and MCF7. This table represents the most potent bacterial strains that are capable to degrade paracetamol, which encoded with M33 and RS2. Phenotypic characterization of bacterial strains was done by using transmission electron microscope, which showed smooth surfaced long chain spores of globose shape, which is a confirmatory of Streptomyces species as shown here in the two pictures of the two bacterial strains. Then genotypic identification of the bacterial strains was done by using 16S rRNA sequencing, which proved that the two bacterial strains which proved the name of the bacterial strains. The first one is called Streptomyces creatomycetes, and the second one is Streptomyces flavofuscus. The two bacterial strains were added on the gene bank and also have an accession number on the gene bank. After that, the phylogenetic analysis was done using Mega 11 software, as shown here in this slide. This is the phylogenetic tree for the RS2 and the second one for the M33 species. This table represents degradation products of paracetamol for two bacterial strains in two different conditions, static and shaking conditions. So, 5 samples were analyzed, 2 samples for each bacterial strain, and 1 sample control, which is paracetamol itself. This data was analyzed and related to paracetamol by knowing the degradation pathway of the paracetamol in Streptomyces species. Also, we have seen the similarity between the degradation products for the two bacterial strains. That's because the two bacterial strains are from the same species, Streptomyces species. So, in some experiments, we have used only one degradation product from one bacterial strain to check the toxicity and to do some experiments. We don't have to use the two degradation products from the two bacterial strains. That's because the similarity between the two species. In vitro MTT cytotoxicity assay was done on the degradation product of the two bacterial strains. In this experiment, the positive control was acetaminophen or paracetamol itself. The negative control was DMSO. That's because this degradation product was dissolved in DMSO. Now, the aim of this experiment is to compare the IC50 of the paracetamol with the IC50 of the degradation products to check the more safe one. As shown here, that IC50 of paracetamol in case of HBG2 cell line is 108.6. However, the IC50 of the degradation product ranges from 119.9 to 200.6, which means that the IC50 of paracetamol is less than the IC50 of the degradation products, which means that the paracetamol is more toxic than degradation products on HBG2. And also for the MCF7, you can find that IC50 of paracetamol is 108.4. However, the IC50 of the degradation product ranges from 285 to 441.7, which means that the IC50 of paracetamol is less than the IC50 of the degradation product, which means that the degradation product is more safe than paracetamol itself on the cell lines. In vivo acute oral toxicity on mice model. In this experiment, 5 to 7 weeks albino female mice weighing 15 to 20 grams were fasted for 24 hours before experiment, then divided into 5 groups randomly, treated with acute single oral dose by using gastric gavage. The experiment lasted for 14 days. Group 1 is a normal control group treated only with saline, and Group 2 is a negative control group treated with DMSO, and Group 3 is a positive control group treated with paracetamol with concentration of 200 mg per kilogram body weight of the mice. This is depending on the pilot study and also on the reference paper. Group 4 received degradation product of sample 1B, which obtained from M33 bacterial strain in a static condition with concentration approximately 200 mg per kilogram body weight also. And Group 5 received extracted 1A, extracted sample 1A obtained from bacterial strain M33 in a shaking condition, also with concentration approximately 200 mg per kilogram for body weight mice. After 14 days of the experiment, blood samples were withdrawn from the mice from retro-orbit, and animals were sacrificed, and liver tissues were homogenized for the analysis. In this graphs, you will find serum liver function tests, ALT, AST, and GGT, which showing significant increase in paracetamol group 3 compared to normal control group, and the negative control group group 2. Biodegradation products group 4 and group 5 showed relatively normal serum levels, liver function test values compared to normal control group 1. Liver tissue peroxidation level was expressed as MDA, showing significant increase in group 4 compared to normal control group 1, and relative increase in paracetamol group and group 5 compared to normal control group 1. Liver tissue catalase antioxidant enzyme activity was depressed in the paracetamol group 3. However, group 4 catalase activity was relatively increased activity compared to the normal control group. Liver tissue total antioxidant capacity TAC showed similar result to catalase. Liver tissue inflammatory mediator marker interleukin 6 and the apoptosis initiator enzyme caspase 9 showed significant increase in paracetamol group 3, and also increase in group 5 and group 4 in comparison to normal control group 1 or the negative control group. The liver tissue oxidative stress enzyme SOD activity was significantly decreased in the paracetamol group result group 3 compared to the normal control group group 1, while group 4 activity was relatively similar to the normal control group and group 5. SOD showed relative increased activity compared to the normal control group. Histopathological examination on liver tissue homogeneity. These pictures represent the 5 groups of the animal model, mice model. Picture number A represent normal control group 1, and picture B represent DEMSU negative control group. Picture number C represent positive control group, which mean the group that's treated with paracetamol. And picture number D represent group 4 with extract 1B, and picture E represent group 5, which represent group extract 1B. Histopathological examination of hepatic tissue sections following 14 days assigned for the acute single oral toxicity testing of paracetamol biodegradation products. Group 1 is a normal control group and group 2 negative control group. Both showed normal liver cells histological structure, picture A and B. The positive control group 3, administered the acute oral toxic dose of paracetamol, revealed moderate dilatation of the hepatic central vein and hepatic sinusoids, in addition to moderate inflammatory changes grade 2, with lymphocyte and macrophage infiltration and few numbers of neutrophils. Fatty degeneration of the peripheral zone, which appeared as intracellular fat droplets. Midzonal coagulative necrosis of hepatocytes and hyperplasia of Kupffer cells were all seen as grade 4, as seen in picture number C. Picture number D represented group 4, M33 extract 1B. There is a mild vascular change compared to the normal group, but not inflammatory case. Group 5, M33 extract 1A showed normal histological structure of hepatic lobules with mild dilated hepatic sinusoids, but no markers of inflammation. In silico study done on the biodegradation products. After the prediction of biodegradation products, molecular structure, solubility and absorption, then distribution, metabolism and excretion also was done by using in silico study. And also functional toxicity prediction was done using Swiss ADME chemo-informatics platform and the BKCSM web tool in silico prediction. Then using both BKCSM web tool and also Swiss DMA.ch. Accessed on 26th of January 2022. This table represents the absorption, distribution, metabolism, excretion, and toxicity of the biodegradation products. The most important thing for us now is that the toxicity of the biodegradation product in silico on liver cells is approximately not present. So now during this project we have reached 2 Streptomyces strains that are capable of degrading paracetamol, and also we have checked the toxicity of this degradation product. We have identified the bacterial strains and deposited these 2 bacterial strains on the gene bank. The recommendation for our work during the coming period is that we need to identify if this novel Streptomyces bacterial strains can degrade estrogenic waste products, and also we have to work on different pharmaceuticals in the environment to check their ability to degrade, to check the ability of these bacterial strains to degrade the new pharmaceuticals. Much.